So this method can help to answer key questions in the macrophage biology field, such as how to characterize their state of activation under various environments at the proteome level. The main advantage of this technique is that it provides the opportunity to obtain differing expression of many proteins in human macrophages using a label-free quantification method. Demonstrating the procedure will be Millet and Magali from my laboratory.
To begin, add 15 milliliters of density gradient cell separation solution into each of two 50 milliliter centrifugation tubes so that the solution can warm to room temperature before receiving the LRSC. This is essential as density depends on temperature. Empty the LRSC into a 50 milliliter centrifugation tube. Add 1 times PBS up to 50 milliliters and mix. Then, very slowly, add 25 milliliters of the mix on top of 15 milliliters of equilibrated density gradient solution. Be careful not to mix the phases during this step. The blood must be added on the density gradient solution without any disturbance of this phase. Centrifuge both centrifugation tubes for 25 minutes at 700 G without breaks. At the end of density gradient centrifugation, the layers from the bottom to top are the erythrocytes and granulocytes forming the pellet, the density gradient solution phase, a layer of PBMCs,